And we're live.
Right. Well, thank you. Thank you, everybody. And we continue our series on God's amazing creation from the book of Genesis, particularly chapter 1. We're on lesson number 8, and we're studying DNA, the life code. And I am with Steve Wilsher, and my name is Tino Unlap. So last week, when we introduced the life code, we stated that it is in every living organism, it is in every cell of every living organism. It is an instruction manual for the making of that organism, what shape it's going to have, the color, even longevity, and the size of that organism is built into that code. And so we say that it is a language that is written using an alphabet that consists of 4 letters, the letters A, T, G, and C. And this code is written in 3.2 billion letters. And so if you were to put all those letters into a book, it will generate 1,000 copies of the Bible in the King James Version. And we also stated from that first study, introducing you all to the code, that if you were to line up all the letters, it will extend, that code will extend into outer space a few billion miles away. And so we concluded that first study by saying the following, that after 6,000 years, when creation occurred, nearly 6,000 years, man finally got the sequence of the letters in that code. This required the work of a number of scientists, groups across the globe, and it took them 13 years to crack the code and $2.7 billion. Now, I want you to think about that as we go through this lesson. It took the creator just microseconds to speak that into existence, into every organism. And yet it took mankind a few thousand years to begin to understand it. And when we focus on elucidating the sequence of the letters, it took us 13 years and a number of groups from all over the world and $2.7 billion. And so for this lesson, we're just going to ask a series of questions and we're going to answer those for you. So let's begin with the first question. Steve?
Well, what does this reveal to scientists?
Okay, well, that's a very good question. So what we see from this code as we learn the sequence of the letters is that it actually is a manufacturing instruction for 20,000 proteins in the cell. Now, let me remind you that we stated that the proteins are the machines in the cell, the cellular machines that actually do the work to make sure that the organism survives. So this is the building or the manufacturing code for 20,000 of those proteins. Now, don't let that fool you. We have over 100,000 of these machines in the cell. So after we manufacture, or the cell manufacture those proteins, they're polished, they're modified to expand the collection of cellular machines even more. And the second thing that it revealed to us is that these only constitute 2% of that code. 2%, that's amazing. And how about the other 98%? Well, you have heard the phrase junk DNA. We used to call it junk DNA until we found that it actually plays a very critical role in regulating how the cell is to manufacture proteins. That's just one example. For instance, heart cells may manufacture different machines because it has a different function from the liver cell or from the kidney cells. And so we used to call it junk DNA because we didn't understand it, but there's a reason why the creator put that in. It's necessary. And so the third thing that we learned from this is that two people that are not related are only different in this code by 0.1%. Fascinating, isn't it? 0.1%, which means that two individuals that are not related are 99.9% similar in a code. You know what's ironic about this, and we will expand on this later on a lesson, and that is, instead of focusing on our similarities, we're 99.9% similar. It's the difference, the tiny percentage difference that we focus so much on that divides us, the human race. Interesting, isn't it?
Yeah.
And interesting. And the final thing that we learned, Steve, is that 98, we are similar to the chimpanzee, 98.8%. And normally, what do people say about that? If we're similar to the chimps, it means people usually say what, Steve? About
We
evolution?
came from the chimp.
Exactly. Well, how about this? If you look at the vehicles that Ford Motor has manufactured, from the most simple to the high-end vehicle, the race that is involved in racing, you will see signature in the engines, right? It doesn't
Right.
mean that one is derived from the other. It just means that they have the same manufacturer.
Designer.
Yeah,
Yes.
designer. And so that's what that is. Really, the fact that we're similar is we have the same designer. So these are some of the very interesting things that we learned. Steve, let's move on to the second one.
Well, tell us how you can get something that is 6 feet long into a cell in a way that you can't even see it.
And that's a very valid question. It's very important. Number one, you have 6 feet, that's 2 yards or 2 meters, right? And you're going to tell me that you can pack that into an entity or into the cell, not just the cell, but part of the cell that we cannot even see with the naked eye. How is that possible? Well, there's an arrangement. Again, the designer designed it in a way that it is very compact in the way that it is packed, and it is packed into the 37 trillion cells that make up the human body. I'd like to play this for you all so you can, they can probably describe it better than I can. Take a look at it.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again.
That's scary. That Steve, that looks almost like a snake. But that
Yeah.
is the way that this code is packed very tightly, very precisely, in a way that it can be so compact that it can fit into a cell that we cannot even see with the naked eye. So let's move on to the third question, Steve.
Well, just tell us about the 3D structure of this code. Explain that to us.
Okay, so just so we don't think that it's just a mash of nothing, right? It's a goo of worthless something. Well, it actually has a three-dimensional structure, and these three-dimensional structure must be maintained in order for the code to be functional. Now, the three-dimensional structure is called helix. It's an alpha helix, and perhaps I can describe it this way. It is like a spiral staircase that has two handrails. And the two handrails are called strands. And those strands are the ones that connect the letters together. So the code is written in letters one after the other in one strand. And then the opposite strand is holding, again, a series of letters that are completely opposite, and we call it complementary to the letters on the one handrail, all right? The strands or the handrails are made up of alternating units of sugar and phosphate molecules. And the steps, the rungs in this staircase, are made up of two letters interacting together to make up one of these rungs. One letter is attached to one of the handrails, and A is always interacting with T on the opposite strand or the handrail, and G is always interacting with C on the other handrail. And so this is a helix, an alpha helix, and it is very similar to a spiral staircase with the two handrails. And there are letters on one handrail that are complementary to the letters on the other handrail. Okay. Next question, Steve.
Well, all right. How is this duplicated?
All right. So as we said, the creator put within each cell of every organism this code. Now, we understand that our cells are made, they grow, they do their function, and they die off. That's just natural. That's natural. They have a lifespan. The cells are made, generated in the cell, in the body, and then they do their work, and then they die off, and they must be replaced. So in order for an organism to survive, a cell must be able to divide. So when you have one cell that contains this code within it, the life code, if it splits into two cells, Steve, that means that each one of the two cells must have identical copies of the life code, right? And so how is it replicated? Well, here's the way it is replicated. There are a lot of, and because we're trying to save time, I'm not going to play this video, but there are a lot of cellular machines and the proteins that actually copy or duplicate the code, right? And the speed is not extraordinary. It would copy, each one of these machines will copy this code about 50 letters per second. 50 letters per second. Now, remember, Steve, we say that each cell contains 3.2 billion of these letters, right?
Right.
So how can we duplicate 3.2 billion if the machines are only working at 50 letters per second? That's going to take us some time. Well, it turns out that instead of just one center where you can begin to copy, on one of these strands containing the code, you can have many centers where you can begin to copy the code. And then they all connected together, right? And so the last thing that I want to say about this duplication is accuracy. It is very important that you begin with a code and you pass it on to the next generation, to the next generation, to the next generation, and you do not introduce any mistakes in it. Because a mistake may mean disease. It may mean that one of these machines is not going to make correctly and it's not going to work. And that's a problem. For the survival of that organism, all of the machines must be made correctly. And the code needs to be intact from generation to generation. And so the accuracy of this duplication process is actually one error in 1 to 10 billion letters. Yeah, wow. Yeah, we call that the vitality of the copying process. And that is literally, it doesn't make any mistake. And the reason for that is, I wish we had time. There are built into that cell machines that will go along the the code, and they will read as you're duplicating it, SSL is duplicating it. If there are errors, there are security mechanisms to correct those errors, to the point where it literally, this copying process does not make a mistake. Incredible, incredible.
Sounds like a language that is self-correcting, as opposed to, I may mispronounce a word, but this language that you're speaking of doesn't mispronounce anything.
That's right.
It duplicates it correctly.
Yeah, and how can we design a system that will be as accurate as that, Steve? Yeah, it's
not possible.
It's not possible. It's not possible. I mean, we make mistakes. Even the transcription, right, of the Bible, they would make mistake even in Hebrew, right? They didn't make major mistakes, but just, the dots and so forth. They may make some minor errors here and there, but there's no mistake in this. The language is copied with perfection from one generation to the other. Yeah, it is by the designer. Almighty. Okay, so let's ask the last question there, Steve.
All right, then how do you go from the code to the manufacturing of proteins?
Oh, it's we can spend an entire year on just looking at the manufacturing process, right? And so let me just go through it a little bit, and then I'll play this video, and then we'll wrap it up with the last slide. And that is, so you have the manufacturing instruction on the DNA molecule. That instruction or that code must be transcribed, sort of like you transcribe a document, right? It must be transcribed into another form. And that form is called a transcript, or it's called an RNA, right? That transcription process occurs in the small part of the cell called the nucleus. That transcript must be trimmed. It must be processed. It must be polished for stability. And then once it's polished and it's ready, it is then shipped out of the nucleus into the surrounding environment within the cell. That transcript is read by machines that will transcribe that from the code in the DNA that is now in the transcript and make the protein according to that instruction.
Wow.
It takes, and I'm going to be very conservative with this estimate, the translation. So you have transcription into an RNA, and then that transcript is translated into a protein. So to go from transcription to translation, it takes several hundred of those machines to do that. It's a very precise process that involves many, many, many proteins, okay?
All within one cell?
All within one cell, all within one cell. And let me just play this video, short video, and then we'll go on to the final slide.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay. Now, Steve, let me just say, the transcription translation process are more elaborate, probably 1,000 times more elaborate than that animation. So that's just for the sake of simplicity that animation is created that way. But there are hundreds and hundreds. We don't know all the proteins that are involved in the transcription process and the translation process. So I'll give an example as we close this and see if you can make a statement after I do this. Let's look at one of the machines, and the machine is called HEXOKINASE. You see that 3-dimensional structure on the right. And the HEXOKINASE is the first machine or the first protein that is involved in the extraction of energy from the sugar. You remember when we talk about the connection of all of God's creation, all living things, they depended on energy from the sun, and the energy from the sun cannot be used unless it's converted to a form that they can all access. And green plants on land or in the sea can make that energy and package it into sugar, and the sugar is called glucose. Now, the first machine, the first enzyme that acts on that so that they can begin the extraction process of extracting the energy from that. And this is a mechanism that is common among all organisms, living organisms. It's called HEXOKINASE. Now, let's focus on just that one, Steve. That one machine or that one protein has to be transcribed. Based on the code on the DNA code for that machine, HEXOKINASE, must be transcribed. A lot of proteins are involved in that process. We don't know exactly how many, hundreds. That transcript is polished, then it is transported out from the nucleus to the cytoplasm, and then it is translated. A lot of proteins or a lot of machines, cellular machines, are involved in that translation process. Then when that machine is made, it's not finished yet. It is polished, it is folded into its 3-dimensional structure, and then it is carried by other proteins to its destination, where it needs to be. Yeah, it's got sensors built into the structure so that it basically knows where it's going, where it's supposed to be. There are signals built into it that destine this protein to the final destination where it's going to do the work, right? So, Steve, but I'll use a very conservative estimate and say, let's say this HEXOKINASE, this machine that initiates this extraction process on the sugar, takes 10 proteins to make it from the code to the machine or to the protein, right?
Okay.
It's going to take Each one of those 10 proteins is going to take 10 proteins for them to be made, right? So we take 10 times 10, that's 100 total proteins that are involved in the reading of the code to the translation process of making it, right?
Okay.
I said that it's not 10, but it's hundreds. It may be 1,000, right? Let's say that we multiply that by 10 or by 100. We're going up to 1,000, 10,000, 100,000. And see what that means is the making of that one single machine will require the action of 100,000 different machines to get it to where it's going to work. Now, how many of us, let's say that you eat sugar, Steve, and you say, well, I do this for energy. We don't think about that, right? We think about the taste, but we eat it because it's the quickest source of energy. And we survive on that. So when you eat sugar, how many of us will wait a week, 2 weeks for that machine to come in and begin the extraction? When you need it, when you're running a marathon or you're hungry or you're starving, right? And you need that machine to do the extraction immediately. Even though it may take 10,000 to 100,000 different machines to get this machine to the point where it can begin the extraction, it doesn't take days, it doesn't take weeks, it doesn't take months. It takes very short period of time, seconds to minutes, to make it. Now, I don't know about you, Steve, but when I look at all the manufacturing processes that we have in this world that are designed by humans, I don't know a manufacturing process that is as efficient or even close to being as efficient as this is. And remember, we're only talking about one machine, right? And we're saying that it may involve the action of up to 100,000 different machines. Now, we stated that the manufacturing of 20,000 of those machines is in the code, DNA code, but after you polish each one of them, you could expand it to over 100,000 of them. So the making of each one of those will take thousands of machines with very precise action that is required for the precise manufacturing of that one protein that is going to initiate the process of extracting energy that you're going to need to survive.
So what you're saying is that when I was taught in elementary school that there were simple single cell organisms, there is no such thing as a simple cell. This is the most complex engine that you and I have ever even heard of or thought of.
Oh no, and the human is probably is the most complex, right? And we have over 100,000, but even the most simple cell, right? Single cellular organism, they may have quite a few of these machines, right? And each one of those machines, each one of those proteins must be made in the same fashion or similar fashion as what we see in humans.
Wow.
And so what does this say about what we have been looking at, Steve? I said that faith and science are not contradictory. And I hope you can see that as you're listening to this lesson. One supports the other. You see, the reason why my faith gets stronger every day, Steve, when I look at something like this, give me the only conclusion that we can draw by looking at this very complex process, Steve. What is the only conclusion?
This did not happen by chance. This was intricately designed by the most powerful mind that exists.
Amen to that. And then through what we can see, we go back to our original lesson. God is glorified. Isn't that wonderful?
Wonderful.
Yeah. Thank you, Steve, and thank you, everybody. And we're going to tie all this together. And I hope you will join us in lesson number 9, Well, we're going to tie this to Genesis chapter 1, back to the first word. Yeah, there's a reason why we're doing all this, and I can't wait until next week to finish up this series. It's exciting. Not that we're going to finish it, but that we're going to tie it all together to the cross. It is beautiful. Thank you. Thank you.
Thank you, Tino.
Thank you.